Good morning, everyone. First of all, I want to thank the Pallies-Osmers-Bacher Disease Foundation for the invitation and opportunity to present our work on human glial cell-based therapies for leukodystrophies, including PMD. Also, a deep thank you to all the patients and families here present, even virtually, for being wonderful children and wonderful parents, and for contributing to make the advances in novel therapies possible in the future. My name is Joana Osório. I am a pediatric neurologist, and I have trained at the University of Pittsburgh Medical Center and then have focused on myelin research in Steve Goldman's lab at the University of Rochester Medical Center and currently at the University of Copenhagen, with the intent to contribute to the advances in cell-based treatments for leukodystrophies and other disorders affecting myelin. And I am doing this talk today in behalf of Steve Goldman's lab. As an introduction to this talk, I want to say that I will cover three main points. The first, I will introduce you to human glial cell progenitor cells and the key experiments that support their use as treatment vectors for Pallies-Osmers-Bacher disease and other leukodystrophies. Then I will discuss the application of glial cell-based therapy in the context of Pallies-Osmers-Bacher disease specifically. And lastly, I will briefly discuss some of the important considerations as this treatment progresses towards its possible clinical application. So what are glial progenitor cells? Glial progenitor cells start to be generated in the second trimester of fetal development, and they persist in the adult brain. They have many processes, as you can see in this picture, and form a network throughout the whole brain, both the white matter and the gray matter of the brain. They are recognized by the proteoglycan NG2 that illustrates their multiple processes in red in this picture of the human brain. These cells constitute about 4 to 5% of the adult brain and make a pool of cells that actively divides throughout the whole life. Some cells remain as progenitors through most of life, while others progressively mature as oligodendrocytes to ensure appropriate maintenance of the myelin sheaths. In the context of demyelination, that is lesions within myelin, such as what happens in multiple sclerosis, these are the cells that migrate towards the lesions to promote repair and by further differentiating as mature oligodendrocytes which produce myelin. We have learned from ours and other studies that human glial progenitor cells can have three fates once they are transplanted. One is to continue and remain as daughter GPCs, and as I mentioned before, they replace each other to ensure that these cells form a continuous mesh throughout the whole brain, both white matter and gray matter. And here the cells are illustrated in red, and some of the cells were labeled with a green dye, and you can see that they are very close to each other and form this mesh-like appearance. Other cells differentiate as astrocytes, that is seen in the image in the middle, and represented here with the red processes by the glial fibrillary acidic protein, or GFAP. Astrocytes in these models are primarily generated within the white matter, but can also be generated within the cortex. Lastly, some of these glial progenitor cells mature as oligodendrocytes, and these oligodendrocytes are the cells that will eventually wrap around the axons and produce myelin, which is of highly interest for Pallies-Osmers-Bacher disease. Here is represented by staining with the myelin basic protein, one of the main proteins of myelin, and the human cytoplasm, forming these long processes that are parallel to the axons. This is a video of a shiver mouse, a model of hypomyelination that is used in our and many groups to assess the ability of cells to engraft and myelinate within an hypomyelinated nervous system. So these mice, they have a mutation within the myelin basic protein, which is one of the main proteins of myelin, which results in significant unsteadiness, which is called ataxia, which you see here by the significant tremor, and also seizures and shortened lifespan, which is limited to about 120 days. Upon transplantation of these mice in the neonatal period with human glial progenitor cells, in this case obtained from human sources from aborted fetus in the second trimester of pregnancy, these cells significantly migrate and expand throughout the whole brain, as seen in red in the figure on the right. Each dot represents a human cell, and after 1 year, the cells are widespread and even denser within the white matter areas, such as the corpus callosum and the white matter of the cerebellum. As these cells sense the hypomyelinating environment, they differentiate as oligodendrocytes and produce myelin, as seen in green in the figure on the left. All this green is myelin basic protein, which is normally absent within this mouse and is whole of human origin, showing the big ability of human cells to produce myelin in an environment where myelin was not present before. As a result of this treatment, this is how the mice that are able to be rescued look after 13 months. So if you remember, these mice normally lived only 120 days. So the mice that are rescued have a significantly, relatively normal appearance, very, very mild tremor, but able to ambulate normally and feed and grow and have a lifespan as normal as mice, as unaffected mice. Although fetal-derived cells reliably mimic human glial progenitor cells because they are not genetically altered, their source is limited and is ethically controversial to use them, and such system would not allow the generation of cells in large numbers that would be required for clinical trials. And for this reason, pluripotent stem cells were used to generate human glial progenitor cells. And who are these pluripotent stem cells? So pluripotent stem cells, it means that it is a cell that can generate any cell within the body. And they can be generated, obtained from cell banks from the inner cell mass of the embryo, or they can also be obtained from nearly any cell in the body, but commonly is obtained from skin cells, and genetically programmed to become a primordial cell that will be able to be differentiated into any cell in the body. And these are called induced pluripotent stem cells, or iPSCs. In our case, we then instruct both ESCs and iPSCs towards glial fate by subjecting them to a long protocol that mimics the exposure of specific molecules over specific time points, similarly to what happens during human brain development and is illustrated in the figure on the right. So the different colors within this fetal brain represent the gradients of different substances and molecules that are critical within certain time points to ensure that the brain develops as it should. And this is what we try to mimic during this protocol. So as you can see now on the left, the cells undergo multiple stages through this long protocol that lasts about 160 days. And they are first instructed to a neural progenitor cell stage that is seen on the second line as the cells express PAX6 and SOX1, and after that they are induced into pre-oligodendrocyte progenitor cells, that is the synonymous of OPCs, or oligodendrocyte progenitor cells, as they express early markers, and then the cells are further mature into glial progenitor cells, and they are finally ready to be used once they are expressing markers such as SOX10 and KX2.2, PDGF receptor alpha, and OLIG2 on the fourth line within the figures. Lastly, these cells can be pushed towards terminal differentiation, and as I mentioned before, to become astrocytes, oligodendrocytes, or to maintain as GPCs. For the transplantation, we keep them as GPCs, as these would be the actively dividing pool that will be able to significantly and widespread engraft the mouse brain. And how are these fetal different, or are they at all different from these iPS-derived cells? We have looked at both cells from fetal origin and iPS-derived GPCs as well as ES-derived GPCs, and it's striking that their ability to populate the shiver mouse brain and to generate myelin, here illustrated in red and green as it was in the previous figure, it's similar to the fetal group. Similarly, on the figures on the bottom, on the left, you can see the red and green, representing green the myelin basic protein, and in red the neurofilaments, which are the axons of the mouse. And the green, which is the myelin, is juxtaposing the axons, showing that is wrapping around the axons as it would be expected, and it becomes significantly compact at later stages. Both image is similar within the fetal and iPS group, referring to A3, A4, and B3. In A5 and B4, you see electron microscopy imaging showing that the ultrastructure of myelin is normal, and in A6 and B5 are showing the nodes of Ranvier, which are normal gaps or channels within the myelin sheets that upon human myelination within the mouse, this organization is preserved and the nodes of Ranvier are normally organized. So now I want to start by making a context within hypomyelinating disorders and how could we use these cells for treatments? First, I want to start by showing you how myelination is impaired in Palliser-Marsbacher disease, as you know, and other myelin disorders using MRI. And for that, we want to start with normal myelination. And that is seen as a progressive increase of signal, which is white in this picture, within the white matter of the brain. And you can see that from the neonate to 5 months to 1 year to 5 years, these white structures are progressively more prominent. And that progression is almost up to adult age by 2 years of age. So the image you see by 5 years, it's considered almost the same as the one from adult age. In hypomyelinating disorders, there is a decrease in myelination as expected for the age. So all the patients, the MRI from patients within this figure on the bottom are from patients with hypomyelinating disorders. And all of them have above 2, so they could be compared to the MRI of the 5-year-old child on the top. And you can see that none of them have the pattern of these hyperintense white fibers that you can see on the top. 2 of the cases are of Palliser-Marsbacher disease, as you can see, the 2 in the middle, and other also of unknown hypomyelinating disorders, which there are still many on the left, and also 18q syndrome on the right, which courses with significant hypomyelination. Broadly, glial cell-based therapy can be applied to many of these hypomyelinating disorders by providing cells capable of populating the brain and mature as myelin-forming cells. In order to proceed, it is thus necessary to understand the biology of each disorder and to homogenize the group of patients as much as possible so that we can better predict the outcomes. So Palliser-Marsbacher is a prototypic pediatric hypomyelinating disorder. As you know, is an hypomyelinating leukodystrophy that affects primarily young boys. It's incurable and there's no treatment that is available. It's an X-linked disease. It's caused by mutations within the PLP1 gene, and often these mutations most commonly are duplications, but can also be point mutations or null mutations. And it has 3 major forms: a conatal, more severe form, a classical form, and the spastic paraplegia type 2 or transitional or milder form. It is a rare condition, as you all know. It affects about 1 to 100,000. But it's a prototypic case of a larger case of hypomyelinating disorders, which we don't know the exact incidence, but they are more common if we think about them as a group. There has been a past trial, which is very important, with neural stem cells that went through a phase 1 study that taught us very important points in order to think about the second study from here on. There is a tight patient advocacy group which would help with the organization and eventual recruitment for a trial, and is a disease that is largely limited to oligodendrocytes, which is very important. The fact that other cell types are not as affected as the oligodendrocytes. There is significant genetic heterogeneity of Palliser-Marsbacher disease. The disease, as I've mentioned before, is caused by mutations in the PLP1 gene. And among these 3 different types of mutations, one of them is the point mutations. And here, the point mutations that have been described up to when we released this chapter within the Handbook of Clinical Neurology, 2018, these were the mutations that had been described. And throughout the whole protein, you can see different colors, which means that the red were the conatal or most severe cases, the yellow were the classical cases, and the green were the spastic paraplegia or milder cases. And you can see that there is significant heterogeneity, although there are some points in common, such as the mutations within the transmembrane domains, which are the 4 squares in the middle, tend to be more severe, but it's not always the case. As you can see, also some yellow and green rounds within this area. So in order to design and think about a clinical trial for glial cell-based therapy for Palliser-Marsbacher disease, it is important to have certain considerations in mind, which are things we continue to discuss and to have more input from many people to try to think about all the important points in anticipation. So one of them would be, who are the most appropriate patients in order to think about a trial for PMD? So one point for glial cell-based therapy is that it's primarily a disease, is primarily a treatment for CNS, so central nervous system. So it means to treat the brain, but not the peripheral nervous system of the null cases and spastic paraplegia, because these cells do not cross the barrier from the central to the peripheral nervous system. For this case, the candidates would be primarily conatal or classical phenotypes. Ideally, we would prefer that the patients would have similar mutations or similar phenotypes, and that would be grouped with some kind of genotype-phenotype correlation that would be possible. And this would be important so that we could better predict any possible improvements of the trial, because if the group is too heterogeneous, it would be too difficult to find any effect within the group. Lastly, to use natural history studies will be critical in order to assess the functional outcomes and how the patients are doing in compared to untreated patients. Another point are the importance of the mouse models for Palliser-Morse-Becker disease. And as I mentioned, although we use the Shiver mouse brain, which is a very important model for hypomyelinating disorders, the Shiver mouse brain does not have the same mutation as Palliser-Morse-Becker disease. And although it's very important and is critical to have the proof of principle to use this therapy, it is important to learn from the specific models of Palliser-Morse-Becker disease. And for that, we're also generating chimeras within the brain from cells from patients with PMD and corrected lines in order to understand what's the degree of replacement of healthy cells to sick cells. Also of importance would be the interaction of the immune system with transplanted cells. As we know that from some of the models that there is significant inflammation within some models of PMD, and how that would affect the effects of cell transplantation is something we need to further understand. In terms of the trial itself, there are important considerations such as what will be the duration of immune suppression. We know from prior trial, it has been done for 9 months, but we know that antibodies have been produced later on, so perhaps a longer immune suppression would be required. Also of question are the dose escalation studies, and what are the right dose is something we don't know yet until we test, so we would need to test different doses. Also the distribution of human glial progenitor cells within the human brain. How will these cells distribute in a brain that is much larger than the one of a neonatal brain? And also, what are the best functional endpoints for us to evaluate the patients in the long term? So these are all considerations that are currently being studied and thought about, and that we would like to have as precise answers as possible within the future. So Palliser-Morse-Becker disease has been thought about as a target for human embryonic stem cell derived glial progenitor cells. And the approach would be to use human embryonic stem cell derived glial progenitor cells to do a phase 1 trial, likely with a small number of patients, possibly 6, and with 2 escalating doses that will be defined. The cells will of course be produced under GMP manufacture and with the CMC controls, and there will be significant safety tox studies and efficacy studies that are actually already undergoing within the lab. The surgery dosing, this is the dose that we have thought about potentially so far. And the endpoints primarily would be clinical endpoints with the neurological scales and functional exams, as well as MRI, serial MRIs to assess the evidence of remyelination. So one of the questions we had was how do cells migrate and expand within a larger brain that will be the human brain? And so for that, we have designed a study in the rat adult brain, which added important information because the rat brain is larger than the neonatal mouse brain. And also it allows us this study to address whether an adult host could be still permissive for cells to expand and migrate when compared to the neonatal hosts. Although PMD patients are children, their brain is significantly larger and has a higher amount of white matter, and is thus better represented by grafts in the adult's young rodent brain. We have found, by transplanting the cells at adult age within 1 point in the corpus callosum, that the cells migrate preferentially within the white matter tracts, both within the forebrain and cerebellum, and they continue to expand and migrate up to 1 year after transplantation, as you can see on the image on the right. The red dots are the human cells, in blue is the whole brain, and in green are the white matter structures. And you can see that they migrate extensively over a long period of time, and also they continue to do so up to 1 year after transplantation. And so that was very reassuring for us to think that within an even larger brain, it is expected, or we do hope, that these cells will continue to migrate even larger to the extent of a larger territory that they will have to occupy. So as I mentioned, we have generated in the lab a chimera with both PLP-derived, with Pelizaeus-Merzbacher glial progenitor cells and corrected lines within the same mutation. And here represented in this picture are the PMD line, which is a point mutation corresponding to a severe conatal mutation, which are here in red. And you can see what we call the isogenic correction, which is the same line, but with the correction of that point mutation. And what we learned so far, and this is a study from a postdoc, Pernille Madsen, within the lab, which is still ongoing, is that the healthy cells, which are the green, have a lot more processes. And these processes are the processes that are wrapping around axons and making myelin. So they have a significant advantage to produce myelin in compared to the sick cells. So what this tells us so far is that we think that if we transplant healthy cells in the context of Pelizaeus-Merzbacher disease affected cells, these will likely take over and have a significant advantage to remyelinate. We have now started to produce cells under a GMP facility in order to allow the gross production necessary for clinical trials. And this is ongoing within the lab. The produced cells are frozen and then are being tested to further characterization and more extensive than before, and also tested to evaluate any possible toxicity, and also confirming their myelination capabilities by graphing them within the Shiver mouse brain. Shall these cells behave similar, as we expect so far, these cells can then be used for clinical trials. We have then generated in the lab a pipeline of work that allows the generation of glial progenitor cells from different sources. And as a summary slide, I want just to go over the process. So we can obtain cells from induced pluripotent stem cells, as you see on the top in the figure, from healthy subjects or from patients carrying certain mutations. And by using CRISPR or other means of gene editing, we can either correct mutations or we can insert mutations in cells from healthy subjects in order to study how sick cells and healthy cells behave together. Both these cells can also be obtained from embryonic stem cells from sick patients or healthy subjects with the same strategy. By using these cells, we subject the cells to this long protocol for glia differentiation that I introduced you before. Another alternative that was done in the beginning within the first studies in the lab is to use fetal brain tissue from which we extract and select directly the glial progenitor cells with no need to differentiation protocol. After that, we select the cells of interest by FACS, depending on the cases, or other times we don't need to select because the cells of interest are so robustly present. And we use these cells then to cell transplantation within the Shiver mouse brain or others to generate clinical models that will be important for specific disorders, such as the Shiver mouse brain or the PMD chimera that is currently under study. And we use this information to understand the biology of how the healthy cells replace the sick cells in order to use this information to complete, to have a more complete understanding of what would be the best design for the clinical trial moving forward. So these cells, we hope, that can be important therapeutic agents for many disorders of myelin, not only Pelizaeus-Merzbacher, but as we mentioned, many of the other hypomyelinating disorders, but also other diseases, not only congenital, but neonatal white matter injury, multiple sclerosis and variants, radiation-related white matter injury, vascular leukodystrophies, and leukoencephalopathies, and even neurodegenerative disorders that we have always thought to be more neurocentric, but in which the role of glia has been more and more understood, and can also be important targets and benefit from glia transplantations. So I want to thank you for your attention. And here, this work was both generated in the Center for Translational for Neuromedicine at the University of Rochester and University of Copenhagen. The work has been supported by these multiple agencies, and also OSIGN and Sana Biotech has supported the IND enabling studies for the preclinical trial. I also want to thank to all the Goldman Lab members that are all listed, or active members or members who have left but have contributed significantly to the work here presented, and also members from Micah Niedergaard's lab who work closely within this project, and also our collaborators that are also listed in the slide. So thank you very much for your time. And I am sorry not to see you personally, but I hope that you can see this presentation well. Thank you very much. Bye-bye.